fantastic, right? We're doing some great things with the science and learning so much about disease conditions and how to edit genomes and create cells that might allow us to look and do screening. But there's a whole level of opportunity beyond that, which involves the use of cells that are created for therapeutic applications. So that's what I'm going to talk about now, and a little bit about the considerations. I guess I wanted to ask, how many folks here are thinking about moving forward or bringing technologies into the clinic for a drug type of a substance? You guys are? Okay. And some of the companies here I know are. And are you from one of these companies? Yeah, it's very interesting. So I spent probably the last, 15 plus years at least in this space doing a variety of different cell-based therapies, encapsulated cell therapies, tissue generation, skin, and cartilage, and cardiac regeneration. So I'm very much on the development side, so taking the science and then moving it forward and translating it. And so there are a lot of considerations to do that. When you have to do this, there are many different things to think about. So that's what I'm going to hope to touch on here. And if it's not exactly where you are, that's please ask me any questions you can, but it's kind of be from that perspective of how to move things forward into the clinic. So that's the intent. So this is over here, right? Okay, here we are. So I'm just going to go through what is cell therapy. I think we all know, so we'll probably be able to go through a number of these quickly. Some process flow considerations certainly, but also the things you have to think about when you're taking a technology and translating it, and the regulatory considerations are very important to be able to be ready for a clinical trial and file your investigational new drug application with FDA. Process and scale considerations, very important to think of a lot of these things sooner rather than later. And then some resources essentially that are industry resources that are very helpful and are helping to kind of map the future and influence current policies are there. Okay, that's going backwards. There we go. So what is cell therapy? Cell therapy is the use of cells to move things forward, to fix a disease condition. So we have varying degrees as time has gone on with the way that biotechnology and biopharmaceuticals have generated. So first we have small molecules, of course. They're a lot simpler. They're very easy to characterize. And then we got into a couple of decades, 3 decades ago, probably protein biopharmaceuticals. And then we kind of moved into more antibody technology. So the complexity starts getting higher, the ability to manufacture these things, but moving towards virus, but then into cells, you get a completely different level of complexity that's involved with the characterization of material and understanding and knowing that it's going to be able to give you the therapeutic effect that you want. So there are a number of types of approaches here. So you have autologous approaches, allogeneic approaches. So autologous is coming from your own body. Allogeneic, it would be like a donor that could be given to other people. And there are many different types of cells that can be used here. So stem cells, of course, which would include pluripotent stem cells, mesenchymal stem cells, neural stem cells, hematopoietic. Immunotherapy, so a lot of T-cell based stuff, there's some very exciting work that's going on now in that area. And also in tissue engineering with more or less primary cells that are used in that space. Again, the promise is really expansive. There are many things that people are currently investigating in this space. Again, from the different types of tissues, endoderm, ectoderm, and mesoderm, from a pluripotent standpoint, so generating these from either embryonic or induced pluripotent stem cells, but also from adult stem cells too. So this is where you're getting your blood-based systems, your mesenchymal stem cells, and so on. So adult, pluripotent, and primary tissues, all being used to generate. And the list goes on. Critical limb ischemia, cardiac regeneration, bone regeneration. There's a company here at Bone, of course. ophthalmic indications, right? Macular degeneration is big, neural Parkinson's disease, ALS, Alzheimer's. It's really a phenomenal amount of potential, but it's still very early. Still need to learn a lot about it. And I say that kind of tongue-in-cheek because some of these technologies have actually been around for nearly 20 years, actually. It's from the tissue engineered side of things, right? So this is living skin equivalent. So the concept of tissue engineering, which is cell-based, is actually not that young at all, but the use of some of the newer stem cell technologies certainly are. And if you think back, cord blood transplantation, that's a cell therapy as well. That's been around for quite a long time. So it's building on that knowledge base and moving forward. So the tools that are out there to understand and really break down what the biology is and also characterize these cells to be able to predict what their outcomes are the work that everybody is doing here, and capturing that and moving it forward is where we want to go. So this is just kind of a snapshot of how much is going on out there. It's really quite a bit on a global perspective. A lot of folks are investigating for cell therapies, and certainly in the United States there are quite a few going on. 859 studies. This is probably Wake Forest or Duke, right? They're doing a heck of a lot of stuff. You've got a lot of things happening in the UK, 157 or so trials. Japan, certainly. This is China, of course, 215. So there's a lot of stuff going on there. So it's an industry that's growing, and it's quite an exciting industry. And the reason it's growing is because there's a lot of potential there. So when you have these thoughts, how do you go about doing it, right? I mean, what are the steps involved to understanding or moving something forward? There are, translation is really bringing the therapeutic concept forward and being able to study it in clinical development. So this is basically going into patients with these therapies or technologies. So development, of course, is important, making sure that you're able to generate the cell that you're using that's going to have a therapeutic effect. But part of that is the regulatory challenges associated. They're pretty extensive, right? Because the most important thing that Food and Drug Administration is looking at is safety, patient safety, right? They don't want to have something go wrong. I mean, remember the trials with some of the gene therapy trials where folks, that's basically stalled that industry for 20 years. I mean, now there's some more stuff that is associated with cell therapy that's actually fascinatingly interesting with gene therapy, but we have to be very careful. We don't want to harm anybody, and so that's where the regulatory challenges come in. Process choices, these are critical from the perspective of having something that you can actually manufacture and sell, as a company, as a treatment. You can have the sexiest technology that does wonderful things, but if it costs $500,000 or what have you, and insurance companies aren't going to pay for it, it's not going to be able to be adopted. So process choices and cost of goods are very important, and of course those related to the materials and reagents that you select when you're growing up and manufacturing or generating your therapeutic. So this kind of is an overview of the things that happen along the lines and the development process. So certainly discovery is this is the basic research that and the tools that we use to understand and move forward. This work, of course, goes into preclinical work. So now you're taking a particular cell line that you might have developed or grown and are studying it, but you need to get initial data in a preclinical model to demonstrate that there's a potential use for that from a clinical perspective. So about this time is when you're really saying, okay, I've got something that's working, your data's coming back, but now you have to start preparing for going into the clinic. Challenges that face cell therapy developers, certainly the preclinical data generation again, right? So if you're generating data, you want to make sure that you normalize for everything else, all your experimental variables. You have enough variables or trying to understand how a cell is going to interact in the environment that it's placed on. What are the effects of growth? So senescence, apoptosis. How long can you grow them? Do you have the same cell at P3 that you do at P15, and so on and so forth? So the data that you generate from a preclinical perspective is absolutely critical to the potential demonstration of success in the future. So the materials that you're starting with are key, because you want those to be as uniform as possible, so that doesn't become a variable, so to speak. The data you generate in preclinical leads to your IND filing, investigation of new drug, and then you have to take these processes and do your clinical manufacturing. And it's very, very, very rare that the process you have at the beginning is the same process that you have at commercialization. As a matter of fact, if it is, you're probably in trouble, and I've lived through that as well. We had a benchtop process that was great. This is a company called Organogenesis. They do a living skin equivalent. They're in the Massachusetts area. And there was a wonderful deal with Novartis many, many years ago, and the company was very excited and things were moving. But as sales started to grow, they started losing money faster because the cost to make it was more than the cost that they were selling it. It's an impossible situation. So making sure you think about these things as early as you possibly can is very, very critical. And that goes to the materials, the methods you're using. Because if you're growing mesenchymal stem cells in 2D culture with the serum-based material, and maybe you have some bovine pituitary extract or something like that in there, that's not going to fly anymore. So how do you get that? How do you remove that? And now if you grew something that gave you all this wonderful preclinical data, but you're in a system that can't really be taken for because of adventitious agent concerns, sourcing concerns, what have you, do you have the same cell when you take out the bovine pituitary extract or drop your serum? I don't know. You don't know. How do you know? It's only as good as the glasses you're wearing. The vision that how you're characterizing essentially your cells. So that's a very important piece to think about, and it ties into preclinical. So it's, again, these are the types of materials that are available to you. They're, when you're developing something and taking it down a clinical route, understanding or knowing that you have that potential to move forward with whatever kind of material is very important, and you want to bring something that can be used commercially as soon as you can, really. And so, here's what I was talking about. There's phase-appropriate risk control. You can use these things, and today, the Food and Drug Administration does not say you can't use sera. You can. But do you want to? Is it the most appropriate? It might in certain instances. Nobody's saying you can't, right? So you do phase-appropriate risk control, but the problems with sera, are firstly, it's not a defined component. You're not sure you have, many times you have to screen it. So the cost of having a sera component might be less than a recombinant or a xeno-free material, but you spend a lot of time and effort screening that material. And that market, the cost of that material, it's like a commodity essentially. So it can rise and fall. It's very difficult to count on what it might be for, if there are fires in Australia, for example, your cost of sera might be very, very high one year, and that's very difficult to base a manufacturing process. So, in biopharmaceutics, not necessarily as much in cell therapy yet, they're going to completely xeno-free and completely define serum. We're not there yet in the science here yet, because we don't understand. So this is a very exciting place to be, actually, because we have the opportunity to define these things.
Is that just because of the complexity of the media that are involved, or does it go beyond that?
A lot of it is, it's exactly that. If we could reconstitute sera absolutely the same way every time, which is what different companies, ourselves, but other companies as well, try to do with these xeno-free formulations, there would be no question. The problem becomes the cost that's there, and quite frankly, not completely understanding the science yet. We don't know what are the components that are critical in a bovine serum albumin or a bovine calf serum, whatever it might be, or human AB serum, whatever you're using. We don't know yet, and it's not easy to figure that out. And so that's where the cost of development comes to the companies that are trying to develop these things. But in Europe, for example, and in other parts of the world, there's a lot of regulatory pressure to get away from serum, more so than here in the States. But it's there, right? And so if you have the opportunity to move to a recombinant or a fully defined system, they would prefer that. It would be easier to move through. Otherwise, you might have difficulties. And again, it's all risk-based, right? So this is why this poor lady here is holding her head, because there's a lot to think about. So are you going to use, can you use research use materials? Or are you going to use pharma-grade materials, therapeutic proteins? If you wanted to do a differentiation step and you're going to put dopaminergic neurons into your first patient's brain, what's the quality level that you want to use for that, cytokine that you're using to do that different? You want it as clean as you possibly can. So how do you do that? Well, you can get materials from a company that is in any part of the world, and yeah, sure, it's basic FGF, but how did they make it? What are the components? What are the primary, secondary, tertiary components that might have contact? What about the trypsin that's used to separate the protein from the column that it was used to, right? What's the source, right? That's where the challenges come in. But you need to think of that as you're moving forward. So you really have to back it up. So it's those are the components when you're moving from a translation and bringing it forward. So sterility, of course, endotoxin, of course, mycoplasma. But it's the things that we're just touching on that you really have to know. So having somebody that has that experience is important. Whomever it may be. I'm not saying it's the company we work for, but it could be anyone. Anyone has to do that, and that's what we did all the time too. And again, we talked about that and changing protocols and that sort of thing. So a lot of things to think about. Same vein here. When you are looking for your materials, you want to make sure that the folks have that kind of expertise and understanding of what's required, so that they can have their quality systems that support the material that they're bringing in, so that you can audit those systems and make sure that they affect or are appropriate for what you're trying to do. So these are where the regulatory affairs professionals come in and looking for things like 510K submissions, PMA, of course, at the end of the day. You want to be able to submit a PMA for your project, but in some cases, you're not going to always get that or rarely are you going to get that in a material that you get. Device master files or drug master files, very important. EU, CE mark technical files, Latin America and Asia, Pacific submissions for where these components are coming from, GMP compliance, of course, and then the knowledge of how they're used are very critical when you start moving. So you need to have somebody that has that experience set to be able to make these calls. And again, FDA, EMEA, and the Sino FDA and all kinds of stuff. They're from a global perspective, there's so much that has to be taken into consideration there, because every country kind of has their own things. There's a lot of efforts towards harmonization of these regulatory bodies, essentially, and it's.
Sorry, I have a question. But as a company, I understand you offer products that, so if we ask you for all these things, you have that somewhere in your folder and you can give it to people. But do you also offer support for people that want to start that? Like, because, for example, you are used to all these regulatory affair things. So if I'm a person and I say, hey, I have all these preclinical results and I'm thinking of filing for an IND, do you also offer that as a service?
We offer it as support. I mean, that's why we're here. That's kind of what, there are a few folks in the company like myself who have, come from industry. And yeah, we try and help as much as we possibly can. And we're always available to have a discussion. I mean, sometimes it's hard to catch us because there's a lot. But yeah, that's the intent. I mean, it's not a perfect world.
I realized that, it was something more than just selling product.
No.
You know, I can say, hey, I want something that is GMP compliant, and you say, oh, I have this, but I didn't realize that you also.
We used to be scientists too.
No,
Yeah.
I understand that. But
Yeah.
most of the scientists actually don't know anything about this. So you need to be a special scientist, I think.
Yeah, well, it's not something that you do in an academic setting because you're doing a lot more basic research. But yeah, so we actually take a lot of pride in the fact that we can try and address these types of situations and not understand what it is that you need. And we don't even have everything you would need for some of these. So in other words, if there are so if CRS, we can get as much documentation as we can, but ultimately the person that's using that material has a responsibility, and that's what I meant by sponsor, right? So we don't do the development. We want to work as close to provide, so we want to be as transparent to our sourcing and what we have to be able to say, well, this is what we have, this is what we know. And then the company or group or whomever would do a risk assessment with that information and say, well, the processing has viral inactivation steps, but it's not validated, so it's pretty good, but I'm actually going to be diluting this out, 1,000-fold or 10,000-fold, or by the end, so they'll say, okay, well, we are going to do our own adventitious agent testing on that particular component, but they have to know what's in there or what has been, and that's what we want to do and be able to provide. So we call this CMC ready, so chemistry and manufacturing and controls. There's a section where you describe how you generate or build or make a drug, cell therapy, biopharmaceutical. That's where you describe what you're doing. And we want to make it as easy as possible for these types of things so people can adopt them easier. Process choices, so we touched on this briefly, but if you're thinking commercial, you have to be able to sell it, and it has to be able to be made at a price that people can afford. So two-dimensional plasticware or microcarriers or 3D suspension culture, these are all technologies that you want to think of as soon as you can, because you can grow stuff in dishes or 6-well plates, for example, but are you going to do that when you're supporting, the Alzheimer population in 2050 that's projected to be, 20% of the population on the planet? It's going to be a lot of people if you're successful, right? We all hope that happens, but you can't grow things in 6-well plates anymore. So the same question is, if you grow it in one way, is it the same as the drug that you originally made? So bringing the technologies as far forward so that when you're doing your material generation, you're generating clinical data, which is not cheap at all. It's quite expensive, as a matter of fact. You'll have the least amount of changes, hopefully. So keeping an eye on that. So, you can use outsourcing. We talked about that briefly, because the opportunity, you can do it yourself, but there's so many regulations, sometimes it's easier and cheaper to have somebody else make it. And there are other companies, or a lot of companies that do that. And certain groups, even here in the city, there are a couple of different groups that have cell therapy facilities. And I don't know if they're necessarily for contract. Some of them, MSKCC, for example, they have a cell therapy facility there for early phase, so you can demonstrate proof of principle. And then if somebody picks it up, a big pharma, then they go. And then, commercial scale, it's very important too to start thinking about that and constantly doing process improvements to get to where you need to go to make it cost effective. So there are a lot of people out there thinking about this, right, and trying to do this harmonization. The Alliance for Regenerative Medicine is a relatively young organization here in the United States that works very closely with people at FDA in Washington, with industry, to define, the protocols and define what's required. How do you define an MSc? Is an MSc an MSc? Is an MSc? I don't know. Are they all the same? Depending on what you're looking for your final. They do a lot of work in there. There's funding organization, of course, NISTEM here, CERM in California. The Catapult for Cell Therapy is a UK organization. The same type of thing, trying to understand and what are the requirements, set standards essentially. Alliance for Advanced Therapies is actually a sister organization to the ARM. This is the European, and they're actually merging very soon, probably in the next couple of weeks. Regenerative Medicine Foundation. This is I think that's the one down in Wake Forest in North Carolina, if I'm not mistaken. FIRM, another component, heavily involved with the regulatory approvals in Japan. The IPSC guidelines that have just been approved to allow investigation from a clinical perspective as quick as possible. They're responsible for that. So all these different organizations, CCRN, this is Toronto-based. Go Toronto. Australian Stem Cell Group, and of course ISCT, which is having a big meeting next week. ISSCR, didn't even put that on here, right? So there are a lot of people out there thinking, and the whole idea is to bring the industry forward, and a lot of these people are speaking together. So that's a really good thing, right? Because eventually all these things will, all these different ways of going about things, need to be understood and regulated appropriately. This is just an example of what the Alliance for Regenerative Medicine put out. Communications with the Food and Drug Administration on the development path, how, why, what, when, and where, and that type of thing. So this is the type of thing that these groups put together, white papers, talking about how you do things, the different opportunities, the characterization technologies that are out there. So, again, that idea is all the best thoughts, all the best intent is to move things forward and define it. But eventually, this is kind of where things are, right? So we've got a lot of technologies that are coming out from the basic research and moving into some of these smaller companies. Although these are some of the bigger, smaller companies, but I can shine this over there, and you can put those right here too. Eventually, and what happens is as these technologies move, then you get the interest of the large pharma, and they can bear fruit eventually. So this is not atypical at all. So a number of these coming, Juno right here, coming out of MSKCC, just recently formed. Teva picked up, it used to be angioblasts, but now they're mesoblasts, which are developing a lot of MSC-based products for cardiac, for bone degeneration, many different things. Nordios Janssen here is going on a big spree. So it's taken quite a while for pharma to start gaining confidence, but over the last 5 years, they've been seeing that, hey, there's actually progress being made. So it's an exciting time. And even here in Novartis has actually, short-circuited the whole situation by going directly to UPenn and giving them, $40 million or something. And if it wasn't for the Carl June technology with the CAR-based T-cell stuff, which is fantastic. So there's a lot of scouting, if you will, by big pharma for both academic, but also these spin-outs. So places like this are very, very exciting, very, very important for the generation of these technologies. Just some paper references, newspapers about the CAR T technology that was picked up by Novartis. Carl June, some fantastic results demonstrating, almost complete reversal in various leukemias. Using the CART19 technology. So that's basically a gene therapy of sorts. So they'll isolate T cells and express CART19 using a lentiviral vector, and those gene-modified T cells are then reintroduced back into the patient and recognize leukemia and basically destroy it. But there's a lot of stuff that's been going on. It's a very, very exciting field, and the application of that type of a technology for other types of cancer is being investigated all over the place as well. So there's real progress being made. So it's exciting, but I can tell you that Novartis is very concerned, but what should I say? They're very cognizant of the importance of safety. So they're looking for everything that they can to make sure that they can have a reproducible process that goes all the way back to the beginning again. So it's for Carl June and Bruce Levine for for getting their technology. They're ripping that whole process down to the ground, and they're going to basically rebuild it. But they're going to need to put their Novartis quality, which is, I mean, you can argue it's even higher than FDA, for example, right? I mean, they just take it to the nth degree because they're going to be providing this to the general population. So it's very important. The materials that you choose are critical. Okay, that was my last slide. So that's all I had. So I mean, any questions, I'm more than happy to try and address if I can.
So as a using the model of Novartis, you as a supplier to that whole chain and having the concern of all of the the suppliers of the suppliers of the suppliers, do you also consider building those sorts of relationships with smaller companies that have technology? So for example, I'll talk about myself.
Sure.
I have a recombinant protein expression system.
Yeah.
And one of the types of things we could make in that system are some of the cytokines and things that you might need for stem cell media. It hasn't been done, but if you threw, I don't know, a couple of million at us, we could run 200 or 300 things through and optimize them, and you could evaluate whether that was a good, reliable source of materials for the stuff that you did. I mean, as a general policy, are you guys thinking in that way as well, or
Yeah, I mean, we
reaching down the chain?
absolutely. We look at new technologies all the time. CRISPR technologies, right? Talons before that, right? And so on and so forth. Some of the pluripotent, some of yeah, so we're always evaluating new technologies, but we're not like a venture capital type fund. If technologies are there and ready to go, and that's why the big company is good, because they could take on the rights to license that technology,
No, I mean,
and then
I'm
move it
looking
forward.
For straight contracts. I mean, if you want to invest, I'm still looking for capital,
But-
I'm out there right now getting
You're right.
contracts from people who want to make recombinant proteins for all sorts of things, I mean, mostly vaccines and so forth. But
Right.
this is an area where I know there's an interest.
Yeah.
I've been approached before by stem cell companies, but
There you go. Yeah.
have not actually had any contracts yet. So
Yeah.
pilot projects.
So for yeah, I mean, certainly. I mean, services. We're looking. We work with many small companies. We OEM materials in as well to have them available for other people. There's a whole group that does just that: corporate outreach, in licensing, out licensing, taking on technologies. Yeah, that happens.
I wanted to ask you, your experience is already, went farther away, but you said a lot of these people need to go back to the beginning and rework their protocol. So, which suggestion will you give to, for example, someone that is starting? Like, what will be maintain good manufacture practice, writing all the protocols down? I don't know, probably there are other suggestions, right? To be prepared if one day we're lucky we arrive at a point that we don't have to go back from the beginning. Are there some rules that?
Well, my advice, I'm gonna take it for what it cost you, is just think if you can. I mean, and it's difficult to do so, and you can think about it by having conversations too. I mean, that's part of it. But think about how it would become. What's your end goal? What's your goal? Is your therapy? Are you looking at a therapy? How would that be? So when you're doing your preclinical work and you find a discovery and you're identifying something that could be Parkinson's, well, how many people could be affected? And so that will drive the kind of scale that you have to do. If it's an autologous therapy, scale is less of an issue because you're making a one-off, and that's actually brings a whole other set of considerations because it's very expensive. Now your lot size is a lot size of one, as opposed to spreading the cost out and having many doses, or whatever it might be. So I would say, take a look at where you think you want to go, and if you're thinking about commercial, make sure you think about how that's going to be made, so a process that is going to be scalable, but also the materials that you're used to go in there, and try and get as much information from or talk to people, or go to some of these industry meetings. Facilitate is an excellent one. ISCT is great. You can join and become members of part of these these groups, you can phone us and we can talk through as much as we can, and you break it down. So what are the components you're using now? Well, this one might be, where you're getting it from. So you basically break down your process for what you're making for preclinical and say, well, if I use those materials, can I take them further? If the answer is, I'm not quite sure, but there's other things that work well that are more simpler, more defined, sometimes it's worth it to take those in the early phase development of data, because then that data can be used further, because you'll change less. So kind of look ahead to see where you want to be.
Yeah, you're not revalidating everything once the processes are in place.
Yeah.
Is there any software that deals with that kind of vertical integration? That's quite common in other industries, like people who make Boeing jets or
Sure.
rockets. For them, they've been doing this for 20, 30 years. I'm kind of surprised that we're not more advanced
Well,
in
it's,
the
yeah.
biotech industry with this kind of thing.
I think in parts of it we are. I think protein pharmaceuticals are pretty standard. You've got your CHO, your super CHOs, and all these different media systems that folks use, and you can get your expression titers up by very known methods. But there's always a little bit of not a little, but quite a bit of process development to tune it, right? The components are there, but ultimately the expression, glycosylate, whatever, are going to be critical. The one slide I probably didn't focus as much as I should have on it, but a lot of times, the product is very closely linked to the process. In cell therapy, it's almost saying the product is the process, but not as much in others. So this is part of the nature of the industry, because it actually goes downstream, because the end products are much less defined, and they're adaptive. They're going to do something depending on where you're putting them. They're going to respond to the niche in a cardiac environment, or they're going to respond to osteo signals in an osteo environment, or a critical limb ischemia, right there. And you use it as an MSC. Can you use an MSC? People say they use it for bone regeneration. They use it for immunotherapy, for immunomodulation, and they use it for critical limb ischemia. Well, is that the same MSC to treat all of those? Probably not. So, company X might be working their process and the way they harvest all the way from the beginning, how they get their tissue, how they process it, how they freeze it, the number of times they freeze it. Everything will affect that cell and will upregulate, downregulate particular genes and what have you. And maybe they just found that whatever they were using or studying is good for bone, right? But it might not be for critical limb ischemia, but it's still an MSC. So that it all depends. So what happened, and I think that's where our industry right now is, there's a lot of opportunity, because the characterization of these things and understanding what is critical, and that's why the scorecard and understanding where you are in certain points of stuff are absolutely critical, because you have to characterize and understand what you have. And if you change something up front, how do you know it's changed downstream? Just doing a tri-lineage differentiation are pretty. That's not a very good focus, in my opinion. But what is a good focus? Well, that's what people are working on. So I don't think the industry is quite there to understand all the components to do a total vertical integration, because it's so specific to a particular indication or product or process.
Thank you.
Yeah, okay, you're welcome. Thank you.